Hi, this is introduction lecture to sugars. Since we finished the aldehyde ketone chemistry and we are now able to use some of the chemistry, specifically the acetal chemistry, in understanding the sugar chemistry. So this is, I feel, an appropriate time to study sugars, which is one of the biomolecules, rather than wait until end of this semester when you would probably have forgotten a lot of the acetal chemistry. All right, so carbohydrates occur in every living organism and is essential to life. Under carbohydrates, we have sugar, starch, cellulose, paper, cotton. Those are all considered to be pure carbohydrates. Now, carbohydrates are synthesized by plants during photosynthesis, as you know. So we have in photosynthesis in plants, they take carbon dioxide and water, and if we want to balance the equation, it takes 6 moles of each, and with the aid of sunlight, it makes C6H12O6, and then produces oxygen as the byproduct. All right. Now this C6H6O6 is the formula, or the molecular formula for glucose. And what the plant does is take this glucose, which would be a monomer, and polymerize into polymer, such as starch, cellulose, paper, etc. And that's what we use a lot of times in food, or in using paper, for example. So in us, what we do is a reverse reaction. We take starch, which is a form of a polymer of glucose, and we break it down in metabolism in our body, and we produce carbon dioxide and water. And from this reaction, we get energy out of this. That's what we need to eat. And when we eat, our body metabolizes and gives us energy. So in a really weird, kind of cool way, our energy really comes from the sun, because the energy is really stored in these molecules, and by breaking it down, we're getting the energy. But essentially, when you look back, it comes from the sun, which I think is pretty cool way of looking at it. So what makes these carbohydrates different from each other if all these things are classified as carbohydrates? Okay, so the biggest difference between all the carbohydrates is the size. All right, so we're going to look at some classifications of carbohydrates based on size. All right, so we have the simplest, which are called monosaccharides. These are the simple sugars. That's a P right there, simple sugar. And example of that will be, for example, glucose or fructose, or a lot of names you've heard of. All right. Now, if you go to the next size up, which would be disaccharides, these will be 2 units of monosaccharides chemically linked together. All right, so an example of that would be our table sugar, which is sucrose, and we have other ones that we're going to look at later on. Oligosaccharides are just something in between, really small versus big. So it looks at 3 to 8 units of the monosaccharides. And then polysaccharides are the polymer of these simple units. So they are usually greater than 8 to 10, and they could go, I mean, the size could range from, you know, a couple hundreds to thousands and tens of thousands of these simple sugars, all right? And the examples are starch, and you've heard of glycogen, and cellulose, etc., etc. Okay, so we're gonna progressively start talking about monosaccharides, and then once we've mastered that, we're going to look at disaccharides, and then oligo, and then polysaccharides. So we're going to do it in sequence. So we'll focus our attention on monosaccharides right now. All right, so monosaccharides are further also divided into two classes. One's called aldoses, and the other ones are called ketoses. All right, so it's a very simple classification. Aldoses contain aldehydes, because some simple sugars have aldehydes and some have ketones. All right, aldehyde or ketone, that's how you distinguish. So you look at the structure and then you decide whether it has an aldehyde or a ketone. That's very simple. The second way to classify them is based on the number of carbons in monosaccharide. So if the total number of carbons is 3, then this is called triose. If it's 4, it's called tetrose. If it's 5, it's called pentose. If it's 6, it's hexose, etc., etc. If you look at a structure of a sugar, so for example, down below I have it in a box, an example of a sugar. So notice the CHO is the aldehyde, abbreviation for aldehyde. So if it was a ketone, it wouldn't have that. It would be in the middle here, right? Acetaldehyde O. So this is an aldose, and so we would write aldose for the classification for this question, and then classify by its number of carbon atoms and type of carbonyl group. So you could actually combine the aldose with the number of carbons you have. So this has 1, 2, 3, 4. So 4 is tetrose. So then instead of saying aldose and a tetrose, you could combine them by saying aldotetrose. And that would be a combined terminology. And with that name, we know that it has an aldehyde and a total of 4 carbons. Okay. Now, this question of classifying the sugar as a D or L, let's tackle that problem. All right. I'm going to briefly review the Fisher projection, which you should have gotten in Chem 114. But basically, Fisher projection is drawn with a T, and by convention, when you draw sugars, the aldehyde is always drawn at the top of that T, all right? And then at the bottom is the CH2OH, and then you'll see that the sugars have a lot of hydroxy groups, and you'll have them either on the left or right. All right. So by convention, what has happened is that they looked at the Fisher projection of the D-glyceraldehyde that's naturally occurring, which is what is drawn here, and looked at the chiral center here, and they determined that when the OH is on the right hand side, they gave that designation as D sugars. So if the OH is on the right side, all right, so in this sugar there's only one chiral carbon, so that's not a big deal. But if you look at for example this sugar, what you're always looking at is the last chiral carbon where the CH2OH comes from, and that's where you determine whether it's D or L. All right, so this OH is on the right side as well, so you ignore the other chiral centers. So you're just looking at this when you answer this question, and because OH is on the right, this is a D sugar. All right, now how you remember that is L sugars is if OH is on left. So L with L, left and L, that's how I remember, right? So it turns out in nature, only the D sugars are exist. All right, so L sugars do not exist. All right, so I also want to go back to this, and when you're looking at Fisher projection, you know that the chiral center is in the plane of the paper, and what's coming out at you are the horizontal bonds, and what's going behind you are the vertical bonds. All right, so you're really looking at it like this when you look at a molecule. And remember, when you're looking at Fisher projection, bow tie, because look at this, it looks like a bow tie here, right? So for example, I am going to show you a structure of the glucose in model, and when you are looking at this molecule, here's the cetyl OH. Let me just zoom out. All right, you are looking at each chiral center such that the horizontal bonds are coming out at you, and then in the next carbon, if it's not, then you need to rotate it so that it's coming out at you to determine whether the OH is on the left or right. All right, so you have to do that at each chiral carbon to determine what is on the left and right. Okay, so that concludes this lecture. Oh, and lastly, before I forget, you should be doing these problems after this lecture, 24.1, 2, 7, and 8.